Refactoring refers to the rewriting of computer code to improve it by altering its internal structure without changing its external behavior. In the context of biology, this means altering a set of genes without disrupting their original function. Two scenarios are often encountered. One is genome refactoring. Here, a genome of an organism is rewritten to introduce convenient cloning sites for further investigation or for defining the minimal set of genes for that organism. Such work has been carried out with phage T7, the mycobacterium genome, and yeast chromosomes. The other context is refactoring of genetic devices, often as a prelude to transferring those genes into another organism. In the context of biosynthesis, we discussed how we could refactor a biosynthetic pathway by stripping the enzyme coding genes of their regulation and then recombining them with well-studied parts. We've also looked at some more complex devices involved in the interactions between microbes and other biological systems. But there are also very exotic processes encoded by microbes that have nothing to do with pathogenesis or commensalism. These include biomineralization, DNA manipulation, DNA uptake, spore formation, fruiting body formation, surface adhesion, photosynthesis, chemotaxis, cannibalism, cell geometry, and so forth. In the next few chapters, we will address the questions: can we encode these processes as genetic devices, and can we make new devices on this level of complexity? To explain these more exotic functionalities, let's start with the basics. Every organism minimally needs to solve four issues. They must be able to grow and divide, undergo the central dogma processes, produce the chemicals that enable the central dogma, and have the ability to respond to changes to the outside world. In practice, this requires around 3,000 genes for an autotrophic organism such as a non-pathogenic bacterium, and it is encoded by around 3 million base pairs of DNA. If you compare these genes from one species to the next, you will see they are highly conserved. They are almost identical amongst the enterobacteria, and typically homologs of these genes or alternate solutions are present in all bacteria. Then there is the other stuff, which varies from strain to strain within one species. It is the existence of these secondary functions that often distinguishes one species of bacteria from another and leads to their specialized adaptation to specific environmental niches. These are the genes that encode things like secondary metabolism, specialized developmental programs, virulence factors, and the like. Evolutionary pressure caused by horizontal transfer of these genes pushes their sequence into gene clusters organized at specific regions of the genome. Here you are looking at the Corynebacterium genome. Based on homology to known virulence factors in other organisms, you see the locations of 13 islands. In two other strains of the bacterium, you get similar patches of genes that are present in some strains and not others. This pattern of having conserved loci for a type of gene cluster at specific regions of the genome is very common. For example, if an E. coli strain encodes an O antigen, it most likely will be present between the GND and GALF genes, but will be an entirely different sequence from one strain to the other. The evidence of clustering implies horizontal gene transfer, which implies that somehow these genes are sufficient to encode a process. As we've seen in previous chapters, many things can get broken when you move a set of genes to another organism. One of the challenges in studying the transfer of clusters from one organism to the next is their cost. As of 2013, 10 kb of DNA costs about $2,000, which is non-trivial, and often these clusters are far larger than 10 kb. Additionally, the usual result of trying to transfer one of these clusters into a model organism like E. coli is for something to get broken in the process. Thus, in practice, you rarely can just clone out or synthesize the sequence present in the source organism and put it into E. coli and expect that to work. A specialized definition for refactoring in the devices context is reliable methodology to take a known sequence and then adapt it to work in your choice organism. This is a very new idea in genetic engineering, and very few examples exist yet for these more complex devices. The challenge here is what exactly do you need to take into account to have a robust methodology? The simplest hypothesis interprets a gene cluster as being defined by a set of molecular functions encoded as proteins, a transcriptional control over when each protein is produced, and control of each gene's expression level. If that is all that is required to have a well-performing gene cluster, then it should be possible to remove all endogenous regulation by removing the 5' and 3' UTR sequences, reshuffling the codon usage, then putting back appropriate ribosome binding sites and promoters. It is not clear yet how general this approach is, but time will tell. We're going to stick to simple prokaryotic processes and explain how a few work. But I want to show you this video so that you can see just how complex microbiological devices can be. This single-celled organism is called Bursaria. If you look at many of these organisms under a microscope, you'll see they are all structurally identical. They each have equally sized feeding organs that siphon in food particles and then digest them. If this were a worm, it wouldn't be anything extraordinary. Multicellular organisms have things like mouths and stomachs ubiquitously, but this is a single-celled organism. There is no intermediary modular object inside this one cell. These are in the realm of chemical objects, and all this morphology must be encoded as biochemical interactions. We won't try to make sense out of Bursaria in these lectures, but clearly the potential for complexity amongst microbiological devices is quite extraordinary.